Okay. Hi. So good morning, good afternoon, good evening, everyone. So my name is Yan. I'm a member of ISPE's webinar subcommittee. I will be moderating this webinar for today. So welcome. So the topic for today's webinar is Pharmacoepidemiology and Drug Safety Best Paper of 2022. Novel methods for pregnancy drug safety surveillance in the FDA Sentinel System. One of ISPE's one of the goals of ISPE's strategic plan for 2024 is to enhance the recognition of pharmacoepidemiology and drug safety as ISPE's journal. So we hope the webinar today will fulfill this purpose. In today's webinar, Dr. Elizabeth Suarez will discuss the paper in which she was the leading author talking about the drug safety surveillance in pregnancy in the FDA Sentinel System. And we also have the pleasure to have Dr. Vincent Lo Re with us. Dr. Vincent Lo Re is the regional editor for the Americas of PDS. So he will tell us how the editorial board of PDS selected this paper, and he will also give us some clues about the review process at the PDS. So to give you an introduction of the two speakers, so first we have Dr. Elizabeth Suarez. Dr. Suarez is an instructor of epidemiology at the Rutgers University. Her research focused on studying the use and safety of medications in pregnancy. And Dr. Vincent Lo Re is an associate professor of medicine and epidemiology at the University of Pennsylvania. He leads a research program that examines the epidemiology of acute and chronic liver diseases in HIV-infected patients. Without further ado, I will hand it over to Dr. Suarez.
Thank you. Jan, do you want to stop sharing this slide and then I will get started with mine? Thank you. Okay, there we go. So hi, everyone. My name is Liz Suarez. Thanks so much for having me here to present on this paper. I'm really honored to have won the Best Paper Award for PDS in 2022. So I'm going to present this work on behalf of my co-authors as well. We were a team from Department of Population Medicine at Harvard Pilgrim Healthcare Institute and Harvard Medical School, and also at CEDAR from FDA, where we implemented this analysis as part of the Sentinel initiative. So let's get into it. Some disclosures. Of course, these views do not necessarily represent the official views of the US FDA. And the funding information from the FDA for this project is also on this slide. And as Jan mentioned, my current affiliation is at Rutgers with the Center for Pharmacoepidemiology and Treatment Science, although I do have ongoing work in collaboration with folks within the Sentinel system. So what's the big idea of this paper? What's kind of the motivation behind why we pursued this work? And my interest lies in monitoring medication outcomes, medication safety outcomes in pregnancy, because we know that pregnant individuals are rarely included in clinical trials during drug development. So any information that we need on teratogenicity or other potential adverse effects, we have to get from post-market data. And so that's data of patients already using the medications, and we're observing their outcomes as they occur. So pregnancy exposure registries have historically been the primary source for this data, but these registries have some limitations. So they often are constrained in their enrollment. They struggle with meeting enrollment targets at times. They also often are only powered to look at the composite of multiple congenital malformations or birth defects. I may use those words interchangeably in this talk. So it limits our ability to find if there's an increased risk for a specific rare malformation that may not get picked up when we group all malformations together, all the different body systems together. So healthcare utilization data which we can think of as insurance claims data or national registry data or EHR data, has been and can be used quite successfully for complementary studies in this area. And predominantly, we've been using it for these targeted cohort studies where we're interested in a certain exposure and a certain outcome or maybe set of outcomes of interest that we may have a prior hypothesis that there's an effect. And what this project aimed to do was thinking about expanding our use of this healthcare utilization data to encompass signal identification methods to supplement these current practices for monitoring. So signal identification broadly is the systematic evaluation for potential adverse events related to the use of medical products without having to pre-specify an outcome of interest. So it allows us to look at a wide range of outcomes and detect new and unsuspected potential safety concerns, and we don't have to focus on a particular outcome of interest that we already have a hypothesis about. So we can use this to identify potential alerts that when we can then target our safety studies to when we want to look at a particular outcome and exposure, instead of doing more of any malformation type of study that doesn't give us as much targeted information. And we do this using this large sample size available in administrative data. So how did this project come about? So I think if you're involved in the society, you're probably somewhat familiar with the Sentinel initiative, but this is led by the US Food and Drug Administration to develop new ways to assess the safety of approved medical products, including drugs, vaccines, and medical devices. So the Sentinel system is a part of this, where we try to answer questions for approved medications using standardized analysis programs and common data model for electronic healthcare data. And what we're using for this project and what we use for a lot of the Sentinel analyses is US commercial and public claims data. So that individual insurance claims for reimbursement for medical services and prescriptions. So the development and adaptation of single identification methods is a top priority within the Sentinel initiative. So we were a work group that were tasked specifically with the application of signal identification methods to this specific area of pregnancy exposure monitoring and for outcome monitoring. And this particular study, we focused on infant outcomes following maternal medication exposure. And given my interest and background in being in studying medication exposures and outcomes in pregnancy using claims data, I led this project with Dr. Judy Mara, also at Harvard Pilgrim, in conjunction with FDA. So let's go back to some methods and background on what we're actually doing. So the signal identification process. First, we have an alert where whatever our signal identification method is, this is an outcome that meets whatever our threshold is for alerting. So we'll describe what that means in a TreeScan scenario once we get to the methods. But essentially, it means that we have a lack of compatibility with a null hypothesis that there's no increase in risk. So we have some indication that there could be an increase in risk. A signal is an alert that has been deemed a potential safety issue. So this is something that we want to move forward with for further evaluation. So these are the steps of this process. First, alert detection, which is primarily what we're going to be talking about today, where we use data mining tools such as TreeScan, which is what we use in this project, to assess a large number of outcomes simultaneously for a single exposure comparison. So we have a single exposure of interest, and now we want to see, is there any alert across this wide span of outcomes that we're interested in? We have the step of alert triage, which is taking an alert to a signal, where we review everything we know about the drugs already. Was this alert expected? We can look at individual patient data, and in our claims data, we look line by line at their individual claims, perhaps in the mother and the infant, to understand, is there another obvious cause for this alert that we see? Or this could also inform our future targeted safety study. So at this point, again, we identify if something is a signal. And then we can implement our targeted follow-up study, where we actually design our very carefully considered observational study, where we're considering our exposure outcome relationship. We're doing things like outcome validation and tailored confounding control to ensure that we are having the most valid, the most valid assessment of this association of interest. So we're not talking so much about the targeted side at this stage today. So TreeScan. This is a method that implements a tree-based scanning statistic analysis. So TreeScan is a freely available software where you can find it at treescan.org, which is on the bottom of the screen there. And so it can be used in pharmacovigilance and pharmacoepidemiology analyses and has been used for this purpose in areas outside of pregnancy. And one of the pros of TreeScan is that it can simultaneously scan for an increased risk across multiple outcomes at the same time. And we can test things up and down a tree, which groups our outcomes either as very specific individual outcomes or aggregates them as we move up the tree, which we'll see an example of in a second. This method formally adjusts for multiple scenarios since we are testing for hundreds or sometimes thousands of outcomes at the same time. And it's compatible with multiple epidemiologic study designs and confounding control methods. And this is important because we don't want to generate a bunch of spurious alerts based purely on the fact that we have unmeasured confounding between our exposure and our referent. So we can use TreeScan in conjunction with propensity score methods to try to limit the number of spurious alerts that we then have to triage and follow up. So the purpose of this case study specifically was to demonstrate how we can do this in real world data using TreeScan to look at a pregnancy exposure using the FDA Sentinel System analysis tools. So what do our results look like in real data? And let's use this opportunity to compare a couple of different analysis approaches and propensity score models to get an idea of what's going to be the best way for the Sentinel system to move forward in routine practice when FDA decides that they have a drug that they'd like to use the TreeScan method for. So let's jump into some methods. So to break down TreeScan, the tree, as I've been mentioning, allows for testing at individual outcomes and related groupings. And the scan allows for adjustment for multiple testing across the tree. So here's an example of the tree, and I'll walk us through this structure. So we are using claims data. So we're gathering our outcomes from ISPE diagnosis codes. Specifically for our purposes, we used ICD-10 because we're interested in being able to use this into the future, as ICD-10 is the coding system that is now used. We limited our tree to the ICD-10 codes that are major congenital malformations and conditions related to gestational length and birth rate. So again, these are the outcomes that are related to the infant and not to the mother. So if you take a look at this tree structure, we have individual ICD-10 codes at the very bottom, level 6, or we can call this the leaf level. And then as we move up the tree, these codes are grouped into larger composite outcomes based on clinical similarities. And we're taking advantage of the natural structure of ICD-10 to do this. You could also build a custom tree if that was of interest. And just to walk you through one branch of the tree. So at the very top, we have all congenital malformations, so a super broad grouping. Then in the next level 2, we break things down into approximately organ systems. So on this branch, in this example, we have musculoskeletal system. We can break things down even further into malformations of the spine and bony thorax. And then as we move down the levels, we break down into very specific malformations in specific regions. And so what TreeScan allows us to do is to test those individual malformations, but also these higher up composite groupings, and each of these nodes, each of these boxes on the tree, you may hear me refer to them as nodes or outcomes. So we can test all of these simultaneously. So as I mentioned, this is a tree-based scan statistic. And just to give you a super brief overview of the actual statistics of the method, we're looking at a single null hypothesis of there's no increased risk across any node in the tree. And this method uses log likelihood ratios, which are calculated based on observed and expected outcome counts. So we calculate our observed outcome counts in our exposed group based on what we actually observed in the data. And this is for every node in the tree. So you have a count for every single outcome. And then we calculated our expected outcome counts using the referent group in some way. We calculate our test statistic, which is a maximum of these log likelihood ratios across all the nodes. And then we use Monte Carlo hypothesis testing to calculate our p-value. And this is the method that also controls for the multiple testing. And what we end up with is a p-value based on the rank of our real test statistic and our randomly generated test statistics under the null hypothesis. And at that point, you choose an alerting threshold. And for our study, we chose anything that was a p-value less than or equal to 0.05 would be considered an alert that should be investigated further. So now into our actual study design. So TreeScan can be used with self-controlled designs or cohort designs. For our purposes, for looking at a pregnancy exposure, we are interested in using a cohort design. We're comparing an exposed group to a reference group. So we need control for confounding. So we're using propensity score methods. And this log likelihood ratio that I just briefly mentioned before can be estimated either using this binomial-based method or a Poisson-based method. And that has implications also for our study design and our confounding control. So we can use the Bernoulli design, and I'll use this terminology as we move forward in the presentation, where it assumes that all outcomes occur uniformly in the population with a fixed probability of belonging to the exposed group. So this works well in a fixed ratio matching situation, because if you have a one-to-one match, you have the same number of exposed and referent group pregnancies in your analysis, and you would assume then that 50% of all the outcomes you observe should be in the exposed group. So that's how we calculate our expected in that setting. And we can naturally use this with propensity score matching. So we're creating that fixed ratio between our groups using the propensity score match. Whereas the Poisson model works a little bit differently, where it uses that referent group to assume the outcomes follow a Poisson distribution based on the outcome rate in that referent group. So essentially, you're applying the outcome rate that you calculate in the referent group to your exposed group. And so we're using this within propensity score stratification, because we can estimate that expected rate within each strata of the propensity score to control for confounding. So those are the two main methods that we're using in this study. So what's the actual case study? I haven't even told you what medications we're looking at yet. So we were looking for multiple reasons to assess a potential negative case study, or something that we expected to be safe and have no alerts. So we're essentially assessing here the potential for false positives. We ended up looking at these two classes of antibiotics, fluoroquinolones compared to cephalosporins. And we are doing this with those two different approaches, with two different propensity score approaches that I just described. With the matching, we did 1-to-1, 2-to-1, and 3-to-1 matching. And then we used 10 strata for propensity score stratification. And then we implemented 3 different propensity score models, because you remember that we're looking at hundreds or thousands of outcomes at the same time. So we can't tailor our confounders and our propensity score method in the same way that we would in a conventional epidemiologic analysis. So based on prior work from Shirley Wang and Judy Morrow and others, we implemented this multi-stage propensity score approach, where first we defined just this general model where we chose variables that we assume would probably be associated with our adverse outcomes and our exposures. And the ultimate idea here is that you would reuse this model for every time that you implement a TreeScan in pregnancy analysis. There's also the option to tailor it a little bit more to the actual exposure, because we do know what exposure contrast we're looking at. So we can add in indications for our exposure. We're doing an active comparator analysis here. So we looked at the different indications for each of those drugs. And then we can just do a completely empirical approach, where we do this high dimensional propensity score approach. We're just selecting variables from the data that are associated with our exposure. And we're doing an exposure-based high dimensional propensity score, again, and not an exposure outcome-based, because we have this huge list of outcomes that we're looking at. So we wanted to compare those 3 approaches. And so we did this using data from the MarketScan database, which is US private commercial claims data. And our population was live birth deliveries linked to their infants. Because we're using ICD-10 tree, we're limited to October 2015 to 2018, which was the data that we had available at the time of this analysis. And the pregnant individuals included in our study had to be 10 to 55 years, and they had to fill one of our 2 medications of interest during the first trimester. Because our primary outcomes are congenital malformations, we're very interested in first trimester exposure. Of course, you could also be interested in exposure in other trimesters as well. So here is a brief schematic of the study design. So we identified deliveries. I don't know if you can see my pointer, probably not, but we identified deliveries based on the live birth delivery event. And then we kind of defined backwards from that. We defined their gestational age backwards in claims. We define an estimated pregnancy start. And then we define our covariates, et cetera, around that period. So we need to require that they have continuous enrollment in their claims data from delivery through a certain period prior to delivery to be able to measure all these things. So that's step one. We wash out other evidence of other delivery codes during that period so that we're capturing or increasing the chances that we're capturing true pregnancy episodes. We look at first trimester exposure to cephalosporins and fluoroquinolones using both dispensings for NBCs and also injections. We excluded exposure to known teratogens in the first trimester, and then we also excluded exposure to the comparator drug spanning back prior to pregnancy through first trimester. So you're exposed to just cephalosporins or just fluoroquinolones in this analysis, not both. And then our outcome window is from delivery to 180 days. And based on the nature of claims data, where we have infant records that infants that aren't necessarily immediately enrolled in the insurance plan at delivery, we look for our outcome codes in both the mother's and the infant's record so that we can potentially increase our capture of these malformations that might be applied to the mother's record while we're waiting for infant enrollment to occur. And I'll point out that there's no required enrollment during this 180-day window for the Bernoulli analysis because we take advantage of that matched structure in the Bernoulli analysis, and we censor the matched set at the time that one in the set loses enrollment. So we are ensuring that they have equal observed time for the outcomes between the groups. It's just not 180 days required for each group. That is a difference from the Poisson analysis where we don't have the advantage of this match structure. So in this case, we are requiring that they have 180 days enrollment. So this is a slight difference in study design that we're comparing because of course this does slightly reduce the numbers that we have in our cohort. And then our confounder adjustment, we looked at pre-existing conditions from 90 days pre-pregnancy through the end of first trimester. We looked at healthcare screening. We looked at indications in these 7 days prior to and including the dispensing date. Okay. Results. So this is just a really brief description of the cohort. We ended up with about 17,000, 18,000 fluoroquinolone exposed in first trimester and 87,000 cephalosporin exposed in first trimester. They didn't differ on age and they didn't differ dramatically on indications, but if you look at these percentages, you can tell that we didn't do a great job of capturing the indications in our claims. So to walk you through this table of the results, what you can focus on is these columns. So what we're really interested in is the observed exposed cases, and then the expected exposed cases. And then we're looking at the alert of a p-value less than 0.05. And then I'll walk you through each of these analyses. So this is that one-to-one matching, propensity score matching in the Bernoulli model with our general model that's not tailored to our example here. And we see that we have an alert for congenital malformations of the larynx. This is, I'm highlighting this alert here. It doesn't meet our significance threshold, but it's grouped under this broader category that did alert. And we see that it does encompass most of the cases, but the split doesn't meet our significance threshold. And then finally, these last three do not meet our significance threshold. And you can see while they have slightly higher ratios of observed to expected to the others that alert, they're based on really small cases. So these aren't meeting our threshold for alert. Then we move into our next model. We're looking at our general plus indications model. So this is when we added in those infection indications for our antibiotic exposures. And we see no alerts in this case. Nothing came up as significant once we included those additional confounders in the propensity score. And then finally, this is our high dimensional propensity score approach. And we see really similar results to the last, where we see no significant alerts popping up. And we see this is that alert congenital malformations of the larynx that came up in that first analysis. And we see that this ratio of observed to expected is attenuated, which may be due to better confounding control. And then we did some sensitivity analyses where we increased the matching ratio. So this is a fixed ratio match. So there's a bit of a risk to increase the matching ratio because you may exclude some exposed patients if you can't find 2 or 3 referent matches for them. but we could also end up expanding our overall sample size, which may increase our power. So we did this, and we did it only with the general plus indications propensity score. And in this case, we had really good overlap in our propensity score distribution. So if you look at the N of the number of exposed cases included, it's not fluctuating that much. So we're not significantly losing a lot of our exposed patients. We may not be losing that much information from our exposed group by increasing our match. And we see pretty much the same results as we saw before. So let's zoom in on this one alert. We have this one alert for congenital malformations of the larynx. So we looked at the claims profiles for this with our FDA work group members, and we decided that these alerts are generally not considered serious, or this specific malformation would generally not require intervention. But more importantly, based on the different propensity score models that we implemented, there's an indication that this alert might have been due to unmeasured confounding based on being only seen in that model where we have very general selection of our confounders for our propensity score model. So based on this, we concluded that there was no need for additional follow-ups. So basically from this analysis, we didn't observe really any spurious alerts, very few, other than this one that was easy to triage. So then the Poisson analysis, this table is set up in a very similar fashion. You have your observed and expected columns. We have our p-value that we look for a threshold of 0.05. First, we have our general model. Again, this isn't matching. This is propensity score stratification. And we see that we have one potential alert of this bicornate uterus outcome, but nothing else is alerting in this analysis. We have a really large observed to expected ratio here and a big split, because our expected case count is so small. Then we have our general plus indications model, and we see the same alert popping up here. And we have our HDPS model, and we see the same alert again. So that's very consistent across our three models for this analysis. So let's look into this malformation that popped up. So this is a rare malformation of the uterus that's typically not observable even or diagnosed until reproductive age. So when we looked into the line-by-line claims, we saw that this is actually an outcome that appeared on the mother's record and not the infant record. And if you remember that we're looking in records for both the mother and the infant, this is kind of something that can happen when you do that type of analysis. So it was easy to triage and say, okay, this is a malformation of the mother. This is not something we would even be able to diagnose in an infant. So this is spurious. This isn't related to the medication. This isn't related to the infant. This is something that pre-existed. We could have done a few things to eliminate the possibility of an alert like this popping up. We could prune the tree a bit more and take out codes that we know wouldn't be diagnosed in infants. We could also try scanning just the infant record. So there's a lot of choices to be made in this analysis. But on the flip side, that triage stage is able to very easily determine that this is not something that needs to be further investigated. So in summary, we found no evidence that fluoroquinolone use in first trimester increases the risk of these outcomes compared to cephalosporins, no alerts other than the ones that we could easily triage. So we don't need targeted follow-up studies in this case. So we do notice slight differences in results by the methods, and I've kind of been highlighting some differences between these methods as we've gone through the presentation. One thing that I haven't mentioned yet, we did a complementary study to this, and there are other studies looking at power of these methods, of the Poisson method and the Bernoulli method for TreeScan. And we have found that the Poisson model has slightly greater power than the Bernoulli model. So that's going to change what things pop up as alerts between these two approaches. The study designs differed between these two. We had different enrollment and requirements, and the different propensity score methods, the matching versus the stratification, also can result in slightly different referent populations, resulting in slightly different expected counts. So I think that accounts for the differences, although the differences were really just that one alert. So I would say that the differences were quite minimal. A disadvantage or a limitation of this analysis is that we were somewhat underpowered to see increases in risk because our exposed group was somewhat small. This was kind of a consequence of using ICD-10 only, but that was of interest to be able to use an ICD-10 tree into the future. And we have the simulation analysis that I mentioned earlier suggests that 4,000 exposed pregnancies is probably a more comfortable spot to be able to observe some smaller increases in risk. We were still able to observe some alerts though. And then use of propensity score stratification, which could be argued results in less fine confounding control than propensity score matching. We didn't find that resulted in many spurious alerts. So we're using an active comparator setting, which of course helps our confounding control in both types of analyses. And this is just another element to think about in the trade-off between using the Bernoulli approach versus the Poisson approach. And we may prefer using the Poisson approach because we didn't see large limitations in using this approach. And we may, as we saw in the simulation paper, have an increase in our power. So I'm happy to take some questions based on this work, and I'll just, before I close out, highlight some other related studies that we have and others have done in this area for those interested. So as I mentioned, we had this simulation study where we're comparing power between the Bernoulli and Poisson approaches, specifically in this pregnancy setting, looking at the incidence levels that we might find for malformations and the sample sizes that are realistic for pregnancy, because it really does differ from the general population where other types of power studies for TreeScan have already been done. So that was published in Epidemiology this year. Thuy Thai is continuing this work where she's expanding the work to look at maternal outcomes. This was just focused on infant outcomes, but of course we're interested in adverse maternal outcomes as well. And so she presented 2 presentations in the last 2 ISPEs, and hopefully publications will be coming soon from that work. And then also Krista Hubrooks and others in their group have done some really nice work, a case study as well, where they focused on drugs with known harm. So that positive kind of case study analysis, they used an ICD-9 base tree with more historical data to be able to capture, enough exposed pregnancies exposed to known teratogens, which is something we didn't feel we were able to do. So it's a nice complement work that was published in American Journal of Epidemiology. So that's all I have. And I think now we're going to turn it over to Vin for some comments before we jump into the Q&A. So I will stop sharing and take it away, Vin.
Thank you. Just for introduction, I'm Vin Lo Re. I'm at the University of Pennsylvania in the Center for Real-World Effectiveness and Safety of Therapeutics and the Center for Clinical Epidemiology and Biostatistics. And I'm the current regional editor for the Americas for Pharmacoepidemiology and Drug Safety. And given that Dr. Suarez's paper was reviewed in our editorial office, and because it was presented at this year's ISPE during the annual meeting of members as the 2023 Ronald Mann Best Paper, I was going to provide just some insights about this award-winning paper. And Jan Lee, who directs the ISPE webinar subcommittee, asked me to give some insights about sort of the publication process of manuscripts at PDS, and to just go through sort of what were the criteria that that we at PDS use to evaluate and score these papers. So just, and I'm only going to take a brief 12 minutes approximately. So just to my grant support and consulting, I don't think any of this has any relation to what I'm going to talk about. So briefly, I just want to highlight to you why we thought this was an award-winning study and really why this study was important. We all really were excited about the use of TreeScan. So I wanted to just sort of amplify some of the points that Dr. Suarez made about the use of TreeScan to detect adverse event signals for medications used in pregnancies. I'm going to highlight some of the interesting methodological aspects of the study. And then I'll talk a little bit about, just reiterate some of the findings, conclusions, and potential what we thought might be important implications in future directions. And then I'll just briefly conclude with what the publication process in PDS is, sort of pulling back the curtain for all of you, and then how these Ron Mann Award each year is selected. Okay, so why did we think that this award-winning study was important? Well, we all thought at PDS that this was that it's critically important to monitor the safety of medications that are used during pregnancy. I think that's evident. But Dr. Suarez highlighted in her presentation that the current methods of monitoring the safety of drugs used in pregnancy have limitations. And she mentioned that there are pregnancy exposure registries, but that they detect congenital malformations really typically as a single composite outcome, and aren't able to look at the breadth of individual outcomes or groups of outcomes. And admittedly, they have enrollment and retention challenges that affect sample size and power. And then there are cohort studies, certainly, but as we all know, that there are potentially challenges in evaluating many adverse events simultaneously. That is an issue in cohort studies, where you want to look at many broad groupings to potentially individual events. And so enter TreeScan, which could be used in electronic health data to identify safety signals for neonatal and infant outcomes, and so allows you to look at a very large number of endpoints, in this case thousands, and sort of screen those to try to prioritize those drug event combinations that are identified as alerts and which might be safety signals. But to date, it has been unclear if the TreeScan can feasibly identify such outcomes after maternal medication use. So to address this knowledge gap, this study sought to assess the feasibility of TreeScan to detect adverse infant outcomes after maternal medication use during pregnancy. We thought this was an important question with potential public health implications. And as Dr. Suarez mentioned, she sought to compare first trimester use of fluoroquinolones to first trimester use of cephalosporins, the rationale being that there have been no definitive human studies suggesting teratogenicity with either of these classes, and that they really wanted to demonstrate minimal false positive signals. So we really thought that the application of TreeScan was very interesting to detect adverse event signals for drugs used in pregnancy. Now, most of you know that traditionally, safety surveillance really is based on voluntary spontaneous adverse event reporting by providers and patients, and that based on those reports, pharmacoepidemiologists can then perform targeted study to evaluate associations between medical products of interest, be they drugs, vaccines, devices, and adverse events. But data mining methods like TreeScan can potentially identify a whole host of candidate drug event pairs agnostic to hypotheses, and so potentially could highlight potential problems warranting studies. These could be serious adverse events, or they could be less serious adverse events to potentially consider in a product's safety profile. But of course, since this is hypothesis generating, you cannot necessarily make causal conclusions from these results, but these could be drug event pairs that could be evaluated in, as Dr. Suarez mentioned, targeted studies. So she mentioned that the TreeScan is a statistical data mining tool, that it could be applied to administrative claims, to electronic health record data, to simultaneously identify this wide range of potential adverse events within a hierarchical tree of outcomes. So could potentially evaluate broad groupings. So, we're going to look at cardiac, we're going to look at malformations, congenital malformations in general, cardiac malformations, valvular manifestations, and then the most specific, let's say, atrial septal defect. So all simultaneously. And she showed this really nice figure. I'm just reproducing it from the paper, that allows you to, look at the most broad, simply congenital malformations here, noted in level 2 for the musculoskeletal system, level 3 for the thorax and spine, level 4, congenital malformations of the spine, and then level 5, kyphosis. So a really broad branches, and level 5, the leaf. And then she mentioned some details that I think we all thought were quite interesting about the versions of the tree-based scan statistic. That there's the Bernoulli statistic that compares the numbers of outcomes in the treatment and the comparator groups with the expectation that the treatment group outcomes should be consistent with the overall group, and the Poisson statistic that compares the observed to expected outcomes, where the expected count of outcomes is generated from the Poisson distribution. And she talked about that the approach to generating the test statistic is to calculate the maximum log likelihood ratio to detect an increased outcome frequency, and then to adjust for the multiple testing that is inherent in the many, what she referred to as node interval combinations that were considered, and to evaluate whether the most likely cluster is statistically significant. That Monte Carlo hypothesis testing was used with an alert threshold that was set at p less than or equal to 0.05. And then they went into the IBM market scan database, identifying mother-infant pairs with at least 391 days of coverage. Really was very thoughtful in the development of 3 different propensity scores, which we thought was really valuable. The general obstetrical comorbidities. The second was what they referred to as the tailored propensity score, which is the general comorbidity propensity score, plus adding infectious indications for fluoroquinolones and cephalosporins, and then the exposure-based high-dimensional propensity score. And they implemented a propensity score matched analysis using the Bernoulli tree-based statistic with each of those 3 propensity score methods, and then the propensity score stratified analysis method using Poisson tree-based statistics with each of those 3 propensity scores. And again, the threshold for alert, p less than or equal to 0.05. And, you know, I'm not going to go through all the details. She just did that, but again, highlighted each of the, in yellow, the 3 different propensity scores. And this identified in the Bernoulli analysis the congenital malformation of larynx. So again, you know, you're going from a whole host of thousands of potential hierarchical outcomes to identify, you know, important alerts. And of course, then she mentioned here that in this case, for the congenital malformations of the larynx, wasn't observed with the general plus indications propensity score or the high dimensional propensity score. So given the lack of alert in those models, and due to the fact that laryngomalacia, which made up the bulk of these congenital malformations of the larynx, generally doesn't require an intervention, this alert wasn't considered for additional follow-up. And then in the Poisson analysis, of course, she mentioned here about the bicornate uterus being mainly a maternal outcome, not a neonatal or infant outcome. And so this alert was also not deemed to be of relevance or require additional follow-up. So to us, methodologic highlights of the study from the exposure outcome standpoint, the fact that they evaluated dispensing and days supply of the fluoroquinolones within the first trimester through 90 days prior, and identify this whole host of tree-based hierarchical outcomes by maternal infant ICD-10 diagnoses from the date of delivery through 180 days later. And then the development of these 3 propensity score models for confounding control, which we thought was a methodologic strength, as well as the 2 TreeScan based method, additional methodologic strengths. And so they concluded that the TreeScan provides safety signal data, potentially could be in a timely manner, that each of these data resulted in single alerts, but that were not deemed to require follow-up after claims review. She highlighted that the tailored propensity score or the high dimensional propensity score were optimal, because those really were the best at potentially addressing confounding by indication. And she highlighted in her comments that we thought was noteworthy, that the Poisson model with propensity score stratification had more power than the other approaches. And, I think personally that the use of TreeScan could be an important method to enhance drug safety in pregnancy. And from my standpoint, while this was really anchored on exposure to look at a whole host of outcomes during pregnancy, it also could be used to scan a hierarchical tree of drugs for a specific adverse event. And I think certainly that would be something in the future that would be of interest. So congratulations and kudos to Dr. Suarez and her entire team. We certainly congratulated her at the annual meeting of members at this past ISPE. But I was asked from the ISPE subcommittee, the ISPE webinar subcommittee standpoint, to, in my role as regional editor for the Americas for PDS, could you comment a bit about the publication process and how exactly you selected this, your group of editors selected this award? So I just wanted to briefly sort of pull back the curtain and give you a sense of what's happening at PDS, which probably is somewhat similar at other journals. So, team of authors submits a manuscript. The manuscript is first reviewed by editor-in-chief Brian Strom, and so he will make an initial editorial decision. Is this within the scope of the journal? Is it of sufficient quality? And if it is not, then he will issue a desk reject. If it is, then he sends it to one of the regional editors, either myself in the Americas, BJ Park in Asia, Olaf Klungel in Europe. We'll then review it, make an editorial decision. If we think it is appropriate in quality, then we will send it to one of our associate editors, who are typically selected from pharmacoepidemiology programs around the regions. Associate editors will then review the manuscript and will have an initial review where they may say this is not really either relevant of methodologic quality of our journal, and so issue a desk reject, or if they feel it warrants, they will send the manuscript out for peer review. We usually look for at least 2 reviewers for each manuscript. When the reviews are returned, usually around 8 weeks, the associate editor will present the manuscript at a monthly associate editors meeting, and you'll see here either they will recommend a decision of reject based on the reviews and their assessment, or they'll recommend a decision of revise. The authors will revise, and at that point, the editors will either make a decision, associate editor, to send the manuscript back out for peer review, typically if it's major revision, or will simply, if it's less major or minor revisions, they will review the critiques, the response to the critiques, and the revised manuscript, and then make a decision either to reject at that point or to accept. So that's just to give you some sense of how papers like Dr. Suarez made it through peer review and publication. I sometimes get asked about the rejection rate. It's been fairly consistent, 59% in 2022, 57% in 2021, 55% in 2020. And then lastly, just how do we select the Mann Best Paper Award each year? So typically around May and June, Wiley will send all of the papers that were accepted in the journal in the prior year to each of the individual offices in Asia, Europe, and the Americas. And then each of the offices will select what they think are the 5 best, either original research or brief research papers from their office. Of note, any papers, including any of the PDS editor-in-chief, regional editors, or associate editors, are not eligible for obvious reasons for this award. And then once those 15 papers are selected, Wiley prepares PDFs, And each of those papers are then carefully, independently reviewed by editor-in-chief Brian Strom and each of the 3 regional editors, as well as the ISPE past president. So this year it would be me as past president. So and last year it was Olaf. So there isn't an extra, but typically it's the whoever is the past president. And of course, those individuals cannot have any, their papers would not be eligible. And then the papers are scored from a 1 to 10 on the following criteria: the caliber of the writing, the originality, the quality of the methods, and the public health impact. A weighted score is calculated for each of the papers. And then Brian, as editor-in-chief, ranks the paper by the scores. We have a meeting to discuss. Highest score wins the first prize. And then we discuss based on the distribution of the next clustering of scores, which should be the honorable mention. So I just want to conclude by, again, congratulating Dr. Suarez and her team for their award-winning paper. Thank Dr. Jan Lee for his asking me to give some remarks about Dr. Suarez's paper, and happy to take questions and with any of the comments that have come up. Thanks, everybody.
All right, thank you, Dr. Lo Re and Dr. Suarez. So let's jump into the Q&A section. So just a reminder, you can type your questions in the Q&A box, or you can raise your hand to ask the questions. So Michelle, I saw your hand, so please go ahead.
Wonderful. Michelle Johnson-Funk at the University of North Carolina. And just to add my congratulations to those of everyone else. This is really an incredibly impressive body of work. I learned a lot, and it was presented with great clarity. So I know that you're going to expand the use of these methods just by virtue of having done this. My question had to do with settings where conditioning on a live birth because of the outcomes of interest might not make sense. And I know that you've thought a lot about conditioning on live birth and some of the consequences of that. And I wonder if you have thoughts in this context for TreeScan. Is it feasible, and how might this approach be adapted for that setting? Thank you.
Thanks, Michelle. Always asking the hard questions. I think, so there maybe 2 ways to look at this question. One is, I think there were some other questions in the Q&A too about could we extend this to non-live birth outcomes and look at spontaneous abortions. This is something also that I think some of these methodological considerations will be clarified when you see the work that Tuitai is doing with the maternal outcomes, because there they had to really struggle with the challenge of having overlapping exposure and outcome windows. So if you're looking at exposure post 20 weeks in pregnancy, and you're also looking at all these different conditions like preeclampsia, gestational diabetes that could be diagnosed in the mother, you have to deal with that kind of time varying effects to not result in bias by the fact that you're selecting some people out because they have the outcome, or because they are lost to stillbirth, or because they're lost to live birth. So they have, in that analysis, we've tried to do some things that were more, I'm not really prepared to really describe it carefully right now, but we were able to kind of look gestational week by week and build the analysis in that way to avoid encountering some of these issues, which I think if you were to apply this to non-live birth outcomes, you could probably take a similar approach, which we haven't explored yet, but I think it's probably a good case maybe to take it further. We're also doing a non-sentinel study where we look at the outcome of stillbirth as the kind of constant that we're looking at, and we scan for multiple different medications occurring prior to the stillbirth that might be associated with the outcome. So that's another way that you can address it. It's kind of flipping the TreeScan approach on its head. And then in terms of live births and not having them and resulting in bias, we think about that resulting in selection bias. There's competing there's competing risks. And I think in the setting of looking at malformations, we'd probably think about the potential for that bias in the same way we would in a targeted outcome study looking at malformations. So we could consider whether or not our exposure is also associated with the probability of having a live birth, or does our exposure referent itself lead to a miscarriage or a stillbirth? And if it's associated with our outcome as well, might we be missing some that could be biasing our results in that way? So I think that would be part of the thinking when you move into that targeted safety study environment, where we're able to tailor more finely to the specific exposure outcome relationships we have. But you might also want to think about it in the sense that, are we missing alerts because of this? So I think it's a great question, and it's not something that we really explored a lot in this project, but it's a good area for future expansion and work to try to make this as robust as possible.
Thanks so much, Liz. That was great.
Okay, so Liz just talked about the non-birth outcomes. So there is a question: could this method be applied to identify the long-term outcomes?
Yeah, I mean, I don't see why not. I mean, your study design will be a bit different. It depends on what ends up in your tree. It depends on how long you want to follow the infants for. But basically, I mean, the tree can be designed to include whatever outcomes of interest that you want. So we took that ICD-10 structure and we kind of pruned it down to those immediate malformation outcomes and preterm birth and birth weight outcomes that we could measure in that 180 days post-delivery. But I think it would just be a matter of extending your outcome window. And maybe that would be a situation where you want to use that Bernoulli method, where you're not requiring that they have follow-up for the entire window. If you're really extending it to childhood, you don't want to require that they have enrollment through childhood, because as we know, at least in US data, you will end up with very small numbers of people if you try to do that. So there's different methodological considerations, but I don't see why not if you have a tree of long-term kind of maternal or child outcomes that you want to consider.
Mm-hmm. Another question: is there a standardized method of determining whether an alert is a signal?
Yeah, there have been other papers published about kind of the iterative approach that can be taken in alert triage. We generally have the method of, as I kind of very briefly outlined at the beginning of the talk, we looked at the known alerts. We did a literature review. We knew what was already out there for our drugs of interest to know what was expected in that sense, any warnings on the label currently, things like that. And the next step is to look at those individual line-by-line claims so that we can fully understand the picture of the cases that we're observing. It could be some cases that it's a miscode. We're really only looking for one diagnosis code associated with the outcome. So like it could be misclassification. That's something that the claims by claims record can help inform. And that's also done by clinical experts as well. So it's not just us epidemiologists looking at that. We have clinical experts who can weigh in on what's happening with these cases. So that's a general outline of the standardized approach that's taken. I think we're at time.
Yep. So Liz, do you still have time to answer the 3 questions here?
Sure.
A few minutes. Yes. Just let everybody know, so the recording of this webinar will be available shortly after we finish in the Education Center of ISPE's website. So next question. Eileen would like to clarify, how is multiple testing accounted for in the selection of alert thresholds?
Yeah, so the multiple testing, it's not done through maybe an approach you might be familiar with, where it's like that Bonferroni correction where you're correcting your p-value for alerting. It's done through the Monte Carlo testing process, where we have a single null hypothesis that's able to adjust for the fact that we have multiple outcomes simultaneously. So I probably won't do a great job describing the technical aspects of that, but if you look in our paper, we cited some original articles from Martin Kulldorff and others who kind of dig in deeper to the actual statistics of how that works. So it's a little bit different. That's why we're not changing our p-value threshold to be much lower than 0.05, like you might be familiar with.
Great. The last question. So do you have any thoughts about removing the codes with conditions specific to the mother? Would the alert have a, yeah.
In that particular case with that bicornuate uterus, when we looked at the line-by-line claims, we saw that every single one of those claims was on the mother's record and not the infant's record. So in the case, if we had, so I think you can think about it in two ways. I think the question is asking about the specific codes. So yeah, if we took that code out of the tree, then we wouldn't see it, of course. But if we had just looked in the infant record and not the mother record, in that instance, we also would not have seen it, because we could see based on that line by line of the infant and mother claims that it was just specifically coded on the mother's record. So yeah, we could have done some things up front that helped us eliminate the need to kind of look at those line by line claims, but this is kind of the trade-off in decision making that you have to make when you're doing kind of a widespread signal detection exercise. How tailored do you want to get? And it's also one of the things that we learn as we go through this type of demonstration project.
Wonderful. Yeah, thank you, Dr. Suarez, and thank you, Dr. Lo Re. Thank you everyone for attending. So our next webinar will be on October 12th. We look forward to seeing you all again by then.